Hello students, today I am going to discuss some questions on a topic called as biomolecules. Now, so today I am going to discuss some questions on a topic called as what? Biomolecules. Biomolecules. So now, without wasting any time, we'll start with the first question. So, in DNA, the complementary bases are, first option, adenine and guanine, second, and as follows, thymine and cytosine. Second option is uracil and adenine, cytosine and guanine. Third option is adenine and thymine, guanine and cytosine. And the fourth option is adenine and thymine, guanine and uracil. So, we need to understand one important concept right here. In DNA, two types of nitrogen bases we are going to observe. Two types of nitrogen bases we are going to observe. One is purine and second one is pyrimidine. One is purine, another one is pyrimidine. So in purine, what are you going to get? One is adenine and guanine. Here cytosine and thymine you are going to get. Okay, so always we need to remember one thing. Complementary basis means pairing basis. Always remember adenine is going to pair with thymine with the double bond. So complementary basis means pairing. Whereas guanine or cytosine is going to pair up with guanine with the triple bond. With how many triple bonds? With how many bonds? Triple bonds. So, what are complementary bases you got? Adenine with thymine, cytosine with guanine. So, we need to check whether these options are available in the given options or not. So, C option, adenine and thymine, guanine and cytosine. So this is the answer for the first one. So in DNA the complementary bases are adenine and thymine, guanine and cytosine. Clear? Now, so the next question, which one of the following once again, okay, second one, which one of the following is an amine group? Which one of the following is an amine group? So options are insulin, progesterone, thyroxine and oxyfurin. So always remember, whenever there will be a presence of amine group, always the naming should end with oxine. Thyroxine, oxine, always it should end with oxine. So let me draw the structure of this. Benzene, iodine, H. So benzene group will be there. Benzene, once again, OH group will be there. Iodine will be there. iodine will be there. Again, they are connected. Again, there will be a benzene ring with CH2, NH2 group will be there, COOH group will be there. So, in this way, so this is a structure, basic structure I am saying. This is the basic structure of thyroxine. This is the basic structure of oxygen will be there at the center. So basic structure of thyroxine. So it is the basic structure of thyroxine. So where you can observe an amine group. So by this you can say that which of the following is an amino hormone? So thyroxine is an amino hormone due to the presence of amino group right here. So now next question. So the next question is, let me go with next question. Green chemistry means such reactions which A are related to depletion of ozone layer, study the reaction in plants, C produce the color during reaction, D reduce the use and production of hazardous chemical. So the green chemistry is nothing but where what you are going to do, reduce the use and production of what? Hazardous chemicals, poisonous chemicals, dangerous chemicals. So green chemistry is nothing but where you are going to reduce the use and the production of hazardous chemical. So answer will be D. So the answer will be D. Now next question. Once again. Next question. Which of the following hormone contains iodine? Contains what? Iodine. Which of the following hormone contain iodine? First option is insulin, second option is testosterone, third option is adrenaline, and the fourth option is thyroxine. So as I have previously wrote the structure of thyroxine, where along with NH2 some of the iodine groups were present which were directly attached to the benzene ring. So without wasting any time, we can directly mark the option D. Okay, which of the following hormone contain iodine? So option is thyroxine. Thyroxine means amine group is also there plus iodine group is there along with the benzene ring. So one more time if you want I can write. So benzene ring will be there. So I wrote a rough structure. OH group will be there. Iodine will be there. O benzene will be there. Where CH2 CH COOH group will be there, which is attached to NH2 group. NH2 group, yeah. So at this place you are going to observe iodines. So what all you are observing? Iodine components. Okay, iodines are right here plus NH2 is there. So that's why we are going to call it as thyroxine. Oxine means amino group will be there. Thyro is related to iodine compounds. Okay, so the option is D. So next question. The segment of DNA which acts as the instrumental manual for the synthesis of protein is. So in DNA which act as an instrumental manual for the synthesis of protein, So you already know that nucleotides and nucleosides are the bonding, something bonding bridge that going to act like in case of DNA, whereas ribose is not concerned. So option is gene. So gene are the responsible for the segment of DNA which acts as an instrumental manual for the synthesis of protein. So whatever we are discussing, it is a biochemistry topic where you are going to study the chemistry reaction that going to takes place in case of biology, in case of human system. Okay, human system. Now, which of the following does not exhibit phenomena of mutarotation? Mutarotation is nothing but where you are going to rotate the hemiacetal groups, where you are going to observe the rotation of hemiacetal groups. Okay, now, so which of the following does not exhibit phenomena of mutarotation? Fructose, sucrose, lactose and maltose. So out of these, in sucrose we cannot observe the mutarotation process because whatever the hemiacetal group will be present in this, that won't be available exhibit in a free form. So if you want to observe the mutarotation which is optically active, it should be available in what? It should be available in free form. So then only you can observe mutarotation. So that's why the option is B. So if you got any doubts, you can ask me. Any doubts? Okay, now let me go with next one. So fructose reduces Stolins reagent due to, as you already know that, so options are asymmetric carbon, primary alcoholic group, secondary alcoholic group, enolization of fructose followed by conversion of aldehyde by a base. That means whenever a fructose is made to undergo a reaction in presence of alkaline, it is going to produce glucose plus mannose, which are aldehyde groups. So the option we can go with is enolization of fructose followed by the conversion of aldehyde by a base. By a what? Base. Next question. Okay. Which one of the following statement is not true regarding plus, that is dextrorotatory lactose? Option A, lactose C12H12O11 contains A2H group. It is correct. B, on hydrolysis lactose give equal amount of D-glucose and galactose. Correct. Lactose is a beta glycoside formed by a union of a molecule of D plus glucose and molecule of D plus means dextrorotatory galactose, and D option is plus lactose is reducing sugar, does not exhibit mutarotation. So the wrong answer is D. So the wrong answer is what? D. Because in lactose, so option A to C are everything is correct, but in case of D, lactose is reducing sugar, does not exhibit mutarotation they have given, but it exhibit what? Mutarotation. So the option B is, once again, let me rub this. So, which one of the following statement is not true regarding lactose means option D. Okay. Now, Which of the following hormone is produced under the condition of stress which stimulates glycogenolysis in the liver of human beings? Insulin, thyroxine, estradiol, adrenaline. So you already know that whenever there will be a stress condition in our body, adrenaline is going to release. So the option is D. So the option is D. Next. D-glucose, electrorotatory glucose reacts with hydroxylamine yield oxime as we discussed in previous thyroxine structure. The structure of oxime would be option A, option B, option C and option D. As there is a plus sign, so the second carbon should have HOH pattern. Here OH pattern is there, so these two pattern is going to wrong, and here HOH pattern is correct, but for the fourth carbon HOH pattern is not there, OH and H pattern is there, so the answer is C. Okay, are you understanding? So here second carbon should have H and OH, and remaining fourth and fifth carbon should have HOH pattern. Here they got OH and H, this will be wrong, and these two are already wrong because OH and H is there, because this is positive dextrorotatory, not negative. Okay, next question. So answer is C. In protein molecule, various amino acids are linked together by dative bond, peptide bond, alpha glycoside bond, beta glycoside bond. So the answer is, already you know that, is linked with which bond? Peptide bonds. So these are the some questions regarding biomolecules. So remaining questions I am going to continue in next class. So if you got any doubts, you can ask me anytime. Okay, so we are here to solve the queries and doubts. Okay, so remaining concepts we will continue in the next class. Thank you.